Hey everyone! In this video, we are going to be working on section 3 of our mega sheet, which is the difference between prokaryotic and eukaryotic DNA. We're going to compare and contrast these two features of the genomes of these organisms. So what I'm going to do is I'm going to create a T-chart in the box for section 3, where we're going to be reviewing information that we had learned about back in unit 2 over how prokaryotic DNA is different than eukaryotic DNA. So let's get started. So in prokaryotes, you typically have a single chromosome that will contain their genetic information for survival. In eukaryotes, they will have multiple chromosomes. This is a great example of where I use this word chromosome to refer to the DNA. Although technically prokaryotes, because their DNA is circular, there's no centromere, but we're just going to refer to the DNA or the genome for prokaryotes and eukaryotes as chromosomes. Next, we had talked about how prokaryotes have a circular-shaped chromosome, whereas in eukaryotes, it's linear shape. It's loose when it's in its chromatin form, but you can lay it out like a string from end to end, whereas with prokaryotes, it's a circle. Next, let's talk about the location. For prokaryotes, it is found in the nucleoid, which is actually just the cytoplasm in the general area where DNA can be found. In eukaryotes, it is inside a membrane-bound nucleus. Now, prokaryotes do undergo some form of organization for their chromosome. In other words, they do slightly package it. So in the case of prokaryotes, they do a process called supercoiling, and that's aided with an enzyme called gyrase. Now, in eukaryotes, it is much more coiled up, and to the point where you can form something that is known as heterochromatin, which is DNA that stays constantly wound and is never expressed. But with eukaryotes, it's not the enzyme gyrase that helps. The DNA is going to be wrapped around these proteins called histones. And that's a way to distinguish between prokaryotic and eukaryotic DNA. Prokaryotes will not have heterochromatin. All their DNA is going to be expressed. But in eukaryotes, there are many segments of DNA that are permanently wound up and will not be expressed. Additionally, eukaryotes have sections of genes that, although they get transcribed in gene expression, don't actually contribute to the overall protein being made. We call those segments introns, and they are going to be removed in the transcription process. Prokaryotes do not have introns, but eukaryotes do have those introns and additional enzymes to remove introns from gene expression process. Another unique feature to prokaryotes is the presence of plasmids. Now, plasmids are external pieces of DNA, and they're usually circular. So it's circular DNA that comes from the environment. And the whole idea about plasmids is hopefully these bacteria will gain some type of superpower to aid in the survival of the bacteria. Now, please understand that plasmids are not required for prokaryotes to exist. Their basic single chromosome has all that information. Plasmids would contain an additional bonus feature, hopefully, to help the organism to survive in different types of environment. These can be from other prokaryotes. These can be from other organisms that just happen to lose their DNA nearby. And these prokaryotes can take these plasmids in. Eukaryotes do not utilize plasmids. First of all, the plasmids, if they're circular, does not incorporate into the linear DNA, but we don't have the machinery to take those plasmids and incorporate them into our own genome. Now, this is how prokaryotic and eukaryotic DNA is different. It's worth putting at the very bottom what they have in common. All DNA is going to be made up of those nucleotides we had reviewed previously, along with those sugar phosphate backbones. And organisms of the same species will have very similar ratios of those nitrogenous bases. So all humans have very, very similar ratios. Even though we are drastically different in our characteristics, our ratios of A to T, C to G are roughly the same. And that's what can act as that unique fingerprint. Now, last thing, all DNA of living organisms is double-stranded DNA, DS, double-stranded DNA. Now, viruses are not considered living, and so viruses can have single-stranded DNA, double-stranded RNA, single-stranded DNA, but for all living organisms, it is double-stranded, and it is made up of the same nitrogenous base pairs that we had discussed earlier today. I hope this video was helpful. Thanks for watching. Bye.